this computer. All right. A lot of things to go through today. Okay, first thing. What's tryptophan? What's that used for? That's the tool for gene regulation. Yeah. Before we start explanations, can you guys tell me why do we need to regulate our gene expression? Anyone? There's a few advantages. First, preserving energy, increase the energy efficiency. Good. What's the second thing? Second thing is the most important one. That means different types of cells can have different functions. Okay, because all of the cells containing the same set of DNA sequences, genetic informations. So if they don't choose to prohibit the expression of a certain gene, they're gonna have the same function. Yeah, you don't want stomach acid in your eyes, huh? You don't want stomach acid in your ear or anywhere else but your stomach. So you have to suppress the gene expression of the protein that synthesizes the stomach acid anywhere else in your body. Okay, so the functions will be differentiated. Okay, that's the main purpose of having gene regulation. So all the tools that we learned is in prokaryotics, okay, in bacteria. Human beings use a different mechanism, okay, that's much more complicated than this, that we're not gonna learn in high school, okay? So just keep in mind, it's not for human, it's just for bacteria, simple stuff. And that's why it happens in the plasmid, okay? No matter what, have you learned? Tryptophan, lactopron maybe? Have you guys done lactopron? Lactose stuff that was on the plasmid as well. Okay, so plasmid is just a small circular DNA that's responsible for the production of a certain protein that have a specific function. It's just the add-on to the nucleoid. Okay, what's the mechanism? There are two main steps. One is suppression or repression, or whatever. And the other one is called attenuation, okay? Most of you can understand the mechanism of suppression, repression, but attenuation is just a disaster, okay? We're gonna simply go through the repression first, okay? What's gonna happen first? Where does that happen? Transcription or translation? Transcription, okay? Your repression happens in your transcription process, okay? That's where your RNA polymerase is stopped, right? At the operator. That's on the DNA, okay? So at first, we have this regulatory sequence called promoter and operator. And we have the leader gene as well, okay? And we have the tryptophans, like the tryptophan encoding genes. Then you have TRP1, TRP2, TRP3. I'm just gonna write TRP, okay? Because it's not relevant to this. When tryptophan is abundant in our body, do we want to produce more? We don't, okay? Because we already had enough tryptophans. So in a tryptophan enriched environment, we're trying to suppress the gene expression of it, okay? How do we do that? We put a suppressor protein, a repressor protein on the operator so that it can stop the RNA polymerase, okay? And one more thing that you guys have to keep in mind, what are we gonna produce in this sequence? Are we gonna produce tryptophan straight away? No, tryptophan is an amino acid. All the transcription and translation processes produce what? Proteins, okay? What's that protein made of? Oh, what's that protein used for? The protein is responsible for the production of your tryptophan, okay? It's just like making a machine that can produce tryptophan in the future. Okay. Keep that in mind. A lot of people have made mistakes during the exam. We're not producing tryptophans. We're producing the proteins that's for tryptophan production. Okay. So what's going to happen first? No matter if we have enough tryptophan in our body, your RNA polymerase is going to bind to your promoter region, right? And what's going to happen? The transcription process will be initiated. Okay. Even though we're going to suppress it later, it will be initiated by a promoter. It's always going to happen. Okay. So in a tryptophan enriched environment, the tryptophan, I'll just write it like the star. Okay. The star or the tryptophan is going to bind to your what? Bind to what? Repressor protein, huh? And what's going to happen if we have a tryptophan bind onto our repressor protein? Come on, guys. Bio exam is the end of this month. Happy October. It's going to change the shape that it binds to the DNA sequence, right? It's going to be activated or inactivated. It's going to be activated. Perfect. So what do you mean by activated? The shape of your active site, or if you want to call that, okay? The binding site is going to be complementary to your operator region, okay? So it binds to the operator, something like this, okay? What's going to happen next? Once your RNA polymerase proceeds to the region on your operator, your transcription process will be terminated. Okay, why? It's stuck. Simple as that. Okay? So this is called your regulatory gene. Okay? And everything after that, what's that called? Structural gene. Okay? Two types of gene combined together, they're called functional genes. Okay, because they have functions. Okay? They're useful. And there's a lot of gene that is not useful in our body. Okay, it just goes for nothing. About 70%. Okay, so only 30% of the genes in our body is useful in code for actual information. And that's relative to our mutation topic as well. If we have constant mutation rate in our non-functional gene, does it matter? It does not matter because it doesn't encode for anything. Okay, if there's something wrong with the functional gene, we're gonna have trouble. Okay, so what's gonna happen if we don't have enough tryptophan in our body? Guys, remember, this is not a 1 or 0 thing. Okay, if the tryptophan level is relatively high, this is gonna happen. Okay, if it's relatively low, it doesn't mean there's no tryptophan in our body. The bacteria is gonna die if there's no tryptophan. Okay, if the concentration is relatively low, that means there's a small chance that the tryptophan is going to bind to your repressor protein, right? What's going to happen? Your repressor protein will be inactivated, right? Once your repressor protein is inactivated, the binding site, the shape of binding site is going to be changed, right? Something like this, so that it combines to the operator region, okay? That means it doesn't have the repressor, the tryptophan on it, okay? Did I make that clear? I think so. Sorry. If the tryptophan concentration is high, that means we have enough tryptophans. That means we don't have to produce more. We terminate the process by stopping the transcription process on the operated region, okay? And vice versa, if we need more, if we have low concentration in our body, we'll have to make it proceed, okay? Simple, it's just like lactopron, but lactopron is the other way around. If you have lactose, that means you have to produce the enzyme that's responsible for lactose digestion, okay? But the general concept is the same, okay? So in this state, your protein is inactivated, okay? Simple, that's repression. Second step called attenuation, okay? Showtime. After your regulatory region, we're gonna have a special sequence called leader, okay? And the leader can be divided into 4 sequences, leader 1, leader 2, leader 3, leader 4, okay? Leader 1, leader 2, leader 3, leader 4. And that's your tryptophan encoding stuff, okay? What's so special about this? Anyone? Why this sequence can help us attenuate the translation process? So what was it?
It can form a heavy root structure that is not assimilated.
Okay, how? Why?
If the
Well, they're gonna form hair loops, either between 2, 3 or 3, 4, right? What determines that? How do I know when 2, 3 hair loops are gonna form? When 3, 4 hair loops are gonna form?
speed of the transcription.
That's the point. And what determines the speed of your translation? Translation, no? Not transcription. What determines the speed of your translation process? How much tryptophan there is, why the concentration of your tryptophan can affect the speed of your translation process. That's the key point. We're almost there. Why?
And the tRNA with the corresponding anti-codon is the premium to the amino acid trip to the sequence. And if there's not enough trip, they can't add it onto the
Yeah, where is that?
leader one.
Leader 1. Okay, that's the key point. Why we can adjust our translation process according to the tryptophan concentration, because our leader 1 sequence is special, okay? Our leader 1 sequence is gonna encode somewhere, okay? Somewhere in the sequence is gonna encode for 2 consecutive tryptophans, okay? What do we have here? What's this? It's a DNA, right? What are we gonna do about this? We're gonna transcribe it, right? Once we transcribed it, it's going to be a mRNA, right? So what do we have on our mRNA? We have codons, right? mRNA is just a bunch of codons connected together, right? Make sense, huh? So in our leader 1 sequence, for example, huh? Let's say it's just UCC, okay? You may have 2 consecutive codons, UCC, that encodes for your tryptophan. Okay, that means 2 tryptophans is needed to form the polypeptide sequence here. Okay, that's gonna affect the translation speed, okay, of your whole process. If we have enough tryptophan in our body, what's gonna happen? That means the tRNA is gonna have the amino acid on the top, right? And it will be easier for the ribosome to find that tRNA and binds to the codons, right? And to form the polypeptide chain easy. But if the concentration is low, what's going to happen? Well, we can find the tRNA, we can find an anticodon, but turns out there's no amino acid on top, right? That'd be bold. Okay. So does that mean we can continue on? No. We have to stay there and wait until the amino acid is ready. Okay. So it's going to take much longer to find the tryptophan that we need for the translation process. That makes the translation of L1 very slow. Make sense? Yeah? I seem so confused. Heartbreaking. Any question? Question time. Nobody has questions? When I was a student, there were two situations that you won't ask a question. One is completely know something. The other situation is that you completely don't know something. Can't even generate a question from your knowledge. I hope you're the first one. Okay. Okay. So if tryptophan is abundant, the translation process will be much quicker. So we can proceed fluently, right? Smoothly. The ribosome is going to land onto your L2 region, right? Leader 2 region, somewhere around here. So this is an mRNA. What's so special about RNA compared with, it's single-stranded, right? So what does that mean if something is single-stranded? It's extremely unstable, right? Yeah, makes sense? If something is single-stranded, what? I don't know, it's just weird. Ignore that, okay? If something is single-stranded, it's going to have mutation. That's one thing. But the second thing is it has the potential to form a complementary thing, okay? Complementary strand, okay? For example, if we have UACCU and we have UACCU here, what's going to happen? The starting sequence is going to be complementary to the ending session of that sequence there, right? And if we have other sequences as well, continue on. And if we leave this mRNA in a cytosol long enough, what's going to happen? I should have write a complementary strand, not the same thing, okay? Not the, okay. Let's just write it again. Let's say A, G, G, U, A, okay? They're complementary to each other. If I leave this in the cytosol long enough, what's going to happen? They're going to attach, right? They're going to bind with each other because they're complementary. They can form hydrogen bonds in between, right? That's how you make a hairpin loop, okay? Because these parts are complementary, they're going to be attached. They're going to stick together, stuck together. That's how you form a hairpin loop, okay? Okay. Back to the mRNA sequence. If the tryptophan concentration is very low here, where your ribosome should be? If your concentration is low, it's going to take much longer to translate your leader 1 region, right? And your leader 1 region will occupy the ribosome for a very long time, okay? Until the 2 tryptophans are found, okay? So if the tryptophan concentration is low, your ribosome is going to stay at the L1 region for a long time. That means you have to leave L2, L3, and L4 in the cytosol and let them be, okay? So what's gonna happen? If the ribosome is here, when L2, like what's the order for transcription? We transcribe from this direction towards that direction, right? So what's first going to be transcribed? We'll finish L1, we can make L2 and make L3 and then make L4, right? So is it possible for us to have L1 being translated and in the meantime, L4 is being transcribed? Is that possible in the bacteria? Why? So why, if not why? There's always a why. Explain it to me. Simultaneously, why? Why our transcription and translation don't happen simultaneously? Because we have membrane organelles. That's the point of having nucleus membrane in our body. Okay? The nucleus membrane segregates to different environments. One's within the nucleus, one's outside of the nucleus. So that's two completely different environments. And you can only finish the transcription process and make the mature RNA and get them absolute 100% ready and send them outside of your nucleus through your nucleus membrane and do the translation process next. Okay? And that's a bit different. That's a little bit different in the bacteria. They don't have any membrane-bound organelles. That means it's just like a huge swimming pool. Everything is just floating in it, right? So if we have something being transcribed, it's highly likely that the tip of your transcribed mRNA will attach to a ribosome and start translation straight away. Okay, because there's no membrane in between these two organelles. It's supposed to be segregated, but there's no membrane in between. So that's why your transcription and your translation process is going to happen simultaneously in the bacteria or in all prokaryotic organisms. Okay? Make sense? Have Joshua told you this? No. Really? Okay. I think he's better than me.
Oh, I didn't.
He has. I
wasn't
believe he
involved-
has.
I wasn't involved when
Okay.
Josh teaching.
Okay, that's right. So you have Catherine or Joshua? Joshua, okay. Joshua is good. Well, I mean, Catherine is good as well. So where we're up to? It can happen simultaneously. So that means it's possible that alpha is being still transcribed when we're doing the translation of your L1 sequence, right? So what's going to happen? This is being occupied by your ribosome, your L1, and the L4 is being occupied by your RNA polymerase in your nucleoid, right? So what's been floating around during this time? Only your L2 and L3, right? So what's going to happen? They're going to be married, right? They're going to be attached to each other. They're going to form a hairpin loop, okay? That is exactly why L2 and L3 is going to form a hairpin loop when the translation process of L1 is slow, okay? It's not charging. For God's sake. All right. Let me see you guys when you're ready. Okay. And if you finished, think about the other situation. What if we have enough tryptophan in our body? What's going to happen? Something interesting is going to happen. The translation speed of L1 will be really quick or slow. Really quick. Sorry, what's going to happen if you do something really quick? You just finished early, right? Hope we can finish early today as well. If you finish the translation of L1, what's going to happen to your ribosome? It's going to move to your L2 for the next step of your translation, right? So it's just like a zipper. If the thing is on your L2 region, is it possible for this region to attach to the other side, to attach to L3? No, it's not, because ribosome is in between. There's no way that the two sequences are going to bind with each other. That's why 2 and 3 is not going to bind if your ribosome have already proceeded to your L2 region already. Okay? So L2 is relatively long. Okay? It's going to take a while for L2 to finish translation, to finish translating. So in the meantime, your L4 has been already fully transcribed. So what's going to happen? What's the situation now? This part has been already translated. Sorry, this part is done. This part is still being translated. And what's been floating around now? And they're going to form a happy loop. Okay? That's a big ass one. That the ribosome cannot proceed continuously. Okay? So if you form a L2 and L3 hairpin, well, it's a hairpin, but it's not as big as a disaster. Okay? Your ribosome can still proceed and continue on with the translation process. But if your L3 and L4 is stopped, is formed a hairpin loop, it's just like a huge stopper. Okay, it's just like a huge stop sign to tell the ribosome you can't proceed any further. Okay, and here we're facing the problem. We have already terminated our translation process. How can we terminate the transcription after this? Think about it. If we can only terminate the translation process, we're going to have no effect on the transcription side, right? So your transcription process is going to just going to go on and on and on and on and on, because there's no signal telling them we can't do any further in the translation. So we're making many useless mRNAs, right? Why is that the case? Is that true? Are we actually making useless mRNAs afterwards? Think about it. If we want to successfully regulate this gene, we have to terminate the transcription as well, right? Otherwise, we're still wasting energies. There's no point of doing that, right? How can we terminate your transcription process while your translation is terminated in the bacteria? Think about it. And people online can be more active as well. Let me hear your voice. I believe we have 5 people. 1, 2, 3, 4, 5. Okay. 1, 2, 3, 5, 6, 7, 8, 9. Okay. We have 14 people today, and I'm the only one who's talking. How? Do we just let it be? Translation. Keep going. Is that something we want? No, it's not. We have to terminate the transcription process while the translation is terminated, right? How do we do that? I've mentioned before, because your transcription and your translation happen simultaneously. Okay, let's draw a diagram here. What's gonna happen if your transcription and translation happen simultaneously? This is your nucleoid, okay? Your RNA polymerase is gonna attach to here, and start the transcription process and make what? mRNAs, right? So here's your mRNA. What's the direction of your transcription? 3 to 5. Yeah, 3 to 5. Yeah. So, what's going to be on this side? Your leader 4 or leader 1?
Leader 1.
Leader 1. That's 2, that's 3. This might be 4, right? Once your leader 1 has been already created or generated, there's a really high possibility that it's going to attach to your ribosome to start the translation, right? So this could be a transcript, this could be a ribosome. So at this part, we're doing translation, sorry. And the other side, we're doing what? Transcription. What's the situation now? If we have enough tryptophan, 3,4 hairpin is going to loop, 3,4 hairpin loops is going to form, and your translation process is going to be terminated, right? If you terminate your process here, what's going to happen? It's just like a huge ass traffic jam, right? And what's going to happen if you want, let's say, if you're going to a city, we're going to use M3, right? And what if M3 becomes a huge car park, huge parking lot that you can't even get onto it? You're stuck there, right? And we give up. I'll just go to city some later tonight. We're not going, or that's not going there now, right? If there's a huge traffic jam, if this part is stuck, this part will be terminated as well. It's only going to be happen in bacteria. Why is that? Because your transcription and translation in bacteria happen simultaneously. If you happen separate, if you have these two steps happen separately, what's going to happen? If something is stuck there, this part is going to continue on. Nothing's going to be affected because there's a membrane between. Okay, there's no cross-docking between them. Okay, but in bacteria, it's different. Okay, if this part is terminated, it's just a huge ass traffic jam. Okay, and this will be terminated as well. Okay, so in the actual exam, if the question asks you why the transcription will be terminated once the translation is stopped, you say, in bacteria, your transcription and translation happen simultaneously. Okay, that's the gold standard answer. Okay, so what's the specific thing is going to happen on your transcription side? Your RNA polymerase is going to detach from your DNA sequence. Okay, attenuation. Any question? Then we're going to move to CRISPR. If you don't have any question, another disaster. CRISPR. I understand it's a relatively new stuff. A lot of the teachers don't even understand this because it's on new syllabus. Okay, but you are the ones going to the exams, not the teachers, right? Unfortunately. Can anyone tell me what CRISPR is? What's that used for? What's the general mechanism of it? Come on. Anything? Antigen?
Antigen.
Oh, antigen. Okay, you scared me. Antigen, okay, gene regulation, gene editing, gene modification. Compare with the enzymatic tool. Why we prefer to use CRISPR? Why? Because it's more accurate. Why CRISPR is more accurate? What two types of, why are you laughing? What are the two types of ends it's going to be created when you use a restriction enzyme cutting the gene? One is blunt end, one is sticky end, right? Which one is better? Sticky ends is better. Why sticky ends is better? Because you have the half of the residual DNA sequence that can be used for what? Proofreading, right? You can only bind to the DNA sequence that's complementary to your sticky ends, right? That's what makes sticky ends accurate. They can bind to a particular target rather than anything. If you just make a blonde end, it can just attach to anything, right? If you want to use blonde end for gene editing, it's going to be extremely inaccurate, okay? Because it can bind to anything, anywhere, right? If you use the sticky end, it can be relatively accurate, but the length of your sticky end is usually 2 to 3 bases. What does that mean? There's still a high possibility that you may bind to something else because the DNA sequence is too long in our body. Okay, we have millions or billions of base pairs in our body. Okay, it's still not accurate enough. How are we going to make it more accurate? We'll make the sticky ends longer, right? If 3 bases not accurate enough, let's make it 10. Let's make it 20. What's going to happen if you have 20 bases forming the sticky end, so-called sticky ends? It's going to be more accurate, right? Because the DNA sequence that's 100% complementary to your sticky end will be really uncommon, right? The longer sticky end you have, right, the smaller possibility that you're going to pair with something random, some random stuff. Okay, it's going to be more accurate. That's why we use CRISPR. Okay, in modern technology, today's researchers, the length of your CRISPR sequence is usually 20 bases or so. Okay, no more than 30, but accurate enough. Okay, but not 100% accurate. Okay, we still make mistakes. Have you guys heard of He Jiankui? No. It's just what's this? Yeah, gene editing. Okay, thank you for the answer. Didn't see that. Sorry. What's gonna happen? First, where's CRISPR found? What's CRISPR's use originally? That's not for gene editing. We don't do gene editing in an environment, in the natural world, right? What's that originally for? That's the self-defense mechanism of your bacteria. Okay, that's kind of the immune system of the bacteria, the E. coli. Okay, because bacteria can use this mechanism to kill the virus DNAs. Okay, so what do we have originally? Let's speak from the very beginning. How are you going to get immune for something? Let's say if you get, how are you going to be immune to COVID? Adaptive immune response. So what's going to happen first? You're either going to be vaccinated or you get a COVID, right? That's how you get immunity. That's the same thing is going to happen on your bacteria as well. If the bacteria want to have the ability to kill a virus, it's going to be contracted first. Okay? And if the bacteria survived after the virus infection, the bacteria will have the RNA sequence or no, the DNA sequence of the virus attached to its nucleoid. Okay? It's just like a memory mechanism. Okay? Once it's survived, it's going to attach its DNA sequence onto it. Okay? It's this exposure. Okay? Perfect. Good. So we have virus 3, we have virus 2, and we have virus 1 because you're not going to just contract the impacted by only one virus, right? We're going to be affected by a lot of viruses. And that's what's going to happen. Why am I having those gaps in between? Because the virus's DNA sequence is foreign. If something is foreign, you don't know the exactly starting point and end point of the sequence. Yeah? If this is something that you're not familiar with, you don't know where it starts and you don't know where it ends. And if I put them all together, tightly located together, you wouldn't be able to distinguish between V1, V2, and V3. Yeah? You have to distinguish between these three different viruses. You need to tell where does V1 starts and where does T2 ends. And if you put them together, that's not something you already know. You don't know when does V1 starts, when does V2 ends. Okay, and if you make the CRISPR complex accordingly, it's going to be much less effective. Okay, so we have to distinguish between V1, V2, and V3 first. How are we going to do that? How can we be able to distinguish between these three different sequences? What's CRISPR called? Clustered, regularly interspaced, short palindromic repeat, right? Have you guys ever heard of that? Okay. The important thing is palindromic repeat. What is palindromic repeat? 上海自来水来自海上, huh? Like it's going to be the same thing if you read it either way. Okay? If it's ATA, and if you read it the other way, it'll be ATA as well, right? That's called a palindromic sequence. If you read it either way, it's going to be the same. That's something that the gene, that the bacteria has already known, right? So we're going to put these palindromic sequences in between the virus DNAs, okay? There'll be palindromic repeat here, there'll be palindromic repeat here, and that's the palindromic repeat here. How is that going to help us to distinguish between different types of viruses sequences? Because palindromic sequence is something that we already know. Okay? So how do we find virus? Anything except your palindromic sequence will be your virus DNA, right? We don't know what's V1, but I know what's palindromic. So everything except your palindromic will be the viruses. And we regularly put palindromic repeats in between the virus DNA. That's why it's called interspaced regularly interspaced palindromic repeat. Okay, we put palindromic repeat in every gap between the different types of virus sequences. So everything except your palindromic repeat will be your virus DNA, right? So the bacteria will be able to recognize the starting point and end point of your virus DNA. Where is that? It's just in between the two palindromic repeats. Yeah? That's why we have palindromics. Okay? It's not for nothing. It's not just for fun. But we're not able to distinguish the between virus and palindromic yet. How are we going to do with it? What are we going to do with it next? We transcribe it because it's the DNA, right? We always transcribe DNA. DNA is useless unless you transcribe it, okay? So even though we make a subsequent mRNA sequence, it's still going to have the palindromic repeat and the virus DNA like corresponding RNA on it, right? So you have the V1 here as well. You have the palindromic here as well. You have the V2, palindromic, V3, palindromic. Okay, here's the thing. This is the time I'll need to distinguish between your virus and your palindromic cell. What's the sequence that I already know as a bacteria? I know virus or palindromic. Do I know the exact sequence of your virus DNA sequence? No, I don't, because that's foreign. Okay, but what's something that I know? Your palindromic sequences, right? So what am I going to do about it? As long as you know the sequence of palindromic, you can make it complementary, right? So I'll make a complementary strand of your palindromic sequence. So another RNA will be generated that is complementary to your palindromic sequence. So what's going to happen next? The complementary strand is going to bind to your palindromic sequence, and what's left now? What's not being paired? Virus. That's how you find it. Okay? So I make a small RNA sequence that is complementary to my palindromic repeat, palindromic sequence. So, and this part will be attached to what? Palindromic or virus, guys? Palindromic, exactly. Because you don't know the sequence of your virus. There's no way you can bind to your virus, okay? So, this will bind to your palindromic sequence. And all the palindromic sequence will be attached and been paired. And what's left now? This part, this part, this part, they're all viruses, okay? Here's something you need to memorize. This is called CRISPR RNA.
Are all the palindromic sequences?
Yeah. There's no way. Yeah, there's no point in making different types of palindromics, right? There's no point. It's just for recognition. And the one on the top is called trace RNA, okay? Tracer is palindromic, okay? So what's left now? We have already accurately targeted the virus sequences, right? By using palindromic mRNAs and its tracer complementaries, right? What's next? I can make Cas9 complex now, right? But here's the problem. The three virus sequences are all together. They're all connected together. Well, it's unlucky to be infected by a certain virus, but it will be extremely unlucky to get all three at the same time, right? I hope I don't get three viruses at the same time. Sorry, most of the situations, you won't need to use all three types of palindromic, all three types of CRISPR complexes, right? You just need one. Either going to be one or two or three, yeah? Because you're only affected by one at the same time. So what do you do about it? You cut it in pieces, right? What's the enzyme used for cutting DNA or RNA sequences? Come on, guys, what's the enzyme? Start with an R or E? Restriction enzyme, good. What's the other name? Endonuclease, good. So your restriction enzyme is going to cut these three complexes apart, okay? Where does that happen? After your palindromic repeat, okay? After your tracer RNA. Here's your cutting point, okay? You cut your gene here, And what's left here? Let's just let's just use this for an example, okay? What's left now? A RNA sequence, CRISPR sequence, and its subsequent tracer RNA, and the virus sequences here, right? And everything else has been cut already, yeah? By endonuclease or restriction enzyme. This is called CRISPR RNA, right? So is it? Sorry, okay. What's this called? CRISPR RNA, right? What's the complex called? CRISPR-Cas9, right? We haven't mentioned Cas9 yet. What's Cas9? That's the enzyme that's for cutting the virus sequences. Okay? Cas9 is a protein that can be seen as a CISID. It's a type of new endonuclease, but more accurate because it uses a more complicated mechanism. Okay? So if we have our Cas9 protein attached, it's going to form a CRISPR-Cas9 complex. That's why this topic is called CRISPR-Cas9. Not just CRISPR, not just Cas9. It's a complex. Okay? They collaborate. So let's distinguish the structure here. What's the RNA sequence on the top that has a little tail on it? What's the name of a sequence? I just mentioned it. Guys, what's the name of this? Tracer RNA, okay? It's down here. CRISPR RNA, okay? CRISPR RNA, thank God. And what's the protein surrounding it? Cas9, okay? And that's completely capable for cutting DNA sequences now, okay? This is viral. That means it's definitely going to be complementary to a certain sequence of those RNA, right? Of the certain virus. So what's going to happen next time when the virus invade the bacteria again? What's the mechanism of virus infection? The virus injects the DNA sequence into your cell, right? And the DNA sequence is going to like transcribe itself, right? Transcribe and translate itself to make more protein codes and just make more RNA, just make more viruses. Sorry, what's going to be in your viruses, in your bacteria now? If the virus invade it again, viral DNA, right? Sorry, let's say it's just your viral DNA. It's going to decoil for transcription and translation, right? When it's decoiled, what's going to happen? We have the little sequence that's complementary to a certain section, right? And that will attach to the viral DNA, yeah? It's going to attach with the RNA, or DNA, sorry. Sorry, it will be something like this, right? This will be your DNA sequence as well, as usual. This will be your usual sequence as well. But in a little section in between, it will attach to your CRISPR-Cas9 complex. Yeah. So that's how your complex target the gene that is going to cut. Okay, they use these sequences for cutting. All right. I know it's a boring topic, but it's definitely going to be on the exam. Okay, the mechanism. So what's going to happen now? If it's an immune system, if it's a cell defense mechanism, we're going to kill the viral DNAs, right? What's going to happen? The Cas9 is going to cut it, right? But Can our Cas9 protein just cut anywhere you want? No. If you cut here, would that be effective? No. Because that's your palindromic. Yeah, if you cut your palindromic sequences, it's not going to do anything about your environment. Okay, so you have to guide your Cas9 protein to cut the appropriate section, which is the viral DNA sequence. What does that? Okay. The Cas9 protein has to know where to cut and what's the thing is used to tell the Cas9 protein where to cut. PAM. Okay. PAM is called proto spacer adjacent motif. What's a motif, guys? You guys, English is better than mine. What's a motif? It's just a symbol, it's just a marker, right? What does it mark? You need to memorize the location very accurately, okay? The motif, the PAM is going to mark just before your tracer section, okay? Just around here. Yes, do you remember the exam question now? Where's that thing? Number 4, right? You need to tell me that's PAM. Because PAM just indicates the sequence right before you trace the sequence, just here. Okay. Where do we cut the what's that called? Where do we cut the different sections just now? Once we have V1, V2, and V3, where do we cut it using endonuclease? We'll cut after the tracer sequence, right? And what's being used now? We use PAM to indicate the location just before the tracer RNA. Okay. So here's your cutting site. So your Cas9 protein is going to cut the DNA this one. Okay. What's going to happen if you cut your DNA here? What's going to happen? What's going to happen if you cut it here? What? What happens if you cut it here? It will be damaged, right? This is your viral DNA. Remember? What's going to happen if you cut your viral DNA in half? It's going to be neutralized. Okay? If you transcribe or translate the 2 sections separately, it's not going to do anything. Okay? It's not an active viral sequences anymore. All right? That's your self-defense mechanism. That's what happens in E. coli even now. Okay? But in human applications, that's a bit different. What's the difference? Think about it. What's the difference between human application and bacterial application? What's the point of having PAM? What's the point of having tracer RNA? We use tracer or palindromic to do what? To help us locate the viral DNA, right? Do we have to locate the gene that's going to be edited in human application?
Yes.
We don't have to. We make that sequence, right? So we have to know it first, and we artificially generate that sequence, right? Yeah. That means we definitely know the starting point and the end point of the sequence, right? So the main difference is for the bacteria, they don't know where V1 and V2 starts or ends. So they have to use a palindromic repeat to locate the accurate position of your V1 starting point and V2 starting point, right? But in human application, we make the V1 or the V2. It's just the sequence that we're going to cut in our human bodies, right? Or in any eukaryotic organisms. Yeah. That's gene editing. We know we're going to cut somewhere and we record the base pair sequences and we make them in a CRISPR-Cas9 complex. So the difference is we've already know the sequence WV1 and V2 in human applications. Okay, what's next? If we have already know that, do we still have to use the tracer RNA to locate it? We don't have to, because we know the sequence already. Okay, so in human applications, we don't need to use the tracer RNA. That's why we make the two separated RNA sequences the same one. Okay, in human application, it's going to be a single RNA sequence. Why is that? Because we don't have to use this tracer sequence to locate UV1. We've already know that. We don't have to locate it. We don't need to find a location, starting point and end point. Okay, so we just make the whole thing. But why is the shape like this still? Because we have to make Cas9 protein active. Sorry, we still have to make the same shape, but this part does not have any actual function anymore. Okay, this is called a single guided RNA, or you can say guide RNA, but you can't say gRNA. That's not the thing. I know it's on the textbook, but textbook is wrong. Never say gRNA, say sgRNA or guide RNA or single guided RNA. Okay, if you use abbreviation, use SG, not G. Okay, stupid textbook. Quiz time. What's the name of Pam? Anyone? What's Pam?
It's based on Jason.
我很欣慰。Sorry, the next time you see a CRISPR question, I hope you remember what happened today, okay? I went through this very thoroughly with you. I hope you understand, okay? Tell me when you're ready. Ready? Good. All right. Any question about CRISPR now? No? Yes? No? Okay. Let's go back to this. Genes and proteins, we've done this. You guys want to go through enzymes with me? Let's just quickly, briefly go through them, okay? What's enzymes? Yes, protein. That's the key point. What's that useful? You guys do chemistry? Anyone? You don't do chemistry? Lower the activation energy of a particular biochemical reaction. Okay? Just make it faster. Just make the reaction faster and easier. Okay? If you don't do chemistry, just remember that. If you do chemistry, it lowers the activation energy of a particular chemical reaction. In biology, biochemical reaction. That's the only difference. So, what's the mechanism? The substrate, which is your reactant, is going to bind with your active site on your enzyme, right? The site on your enzyme is called the active site, and the thing binds to it is called a substrate. Okay? Once they bind to each other, the chemical reaction will be accelerated, will be proceeded. Okay? And there are two main models to describe the fitting mechanism, right? One is called lock and key, one is induced fit. Which one is more like accurate or modern? Induced fit. Okay, what's lock and key? Very self-explanatory, right? The key or the lock doesn't change the shape, right? So it's just a strictly fitting shape between the substrate and your active side, okay? And when they fit, the reaction happens. When they don't fit, they don't accelerate, okay? What about induced fit? That means something can change the shape slightly once it binds to each other, right? So who's going to change their shape? Substrate or your active side? Or both? What's going to change? Whose shape is going to change? Active side. Always remember that your substrate don't change their shape. The only shape is going to change during the exam. Sorry, thank you. The only shape is going to be changed is your active side on an enzyme. Okay, that's the only thing that's going to be changed. And inhibition mechanisms. There are two types mainly. One is called, and the other is called. Come on, fill in the gap. Inhibition mechanism. If I don't want the enzymes to activate and then to activate a chemical reaction, what's going to happen? One is called competitive inhibition. The other is called incompetitive inhibition, right? What happens in the competitive inhibition mechanism? The inhibitor will just occupy the active site, okay? So once the space has been occupied, your substrate cannot bind to the active site, so the chemical reaction cannot proceed, okay? Is that reversible? Guys, that was an easy question. Is that reversible?
Yeah.
Yes, it is reversible, okay? Once the inhibitor leaves the active site, your substrate can go back to the active site again. Yeah? Make sense, huh? It's just like 站着茅坑不拉屎，对不对？它走了之后，你是不是它就又可以了？对吧？What's the other model? What's the other mechanism? Incompetitive inhibition, okay? So we have to introduce another side called allosteric side, okay? Your incompetitive inhibitor is going to bind to the allosteric side rather than your active side, okay? Once the inhibitor binds to the allosteric side, what's going to happen? The enzyme will be denatured permanently. When I say denatured, always permanent effect, okay? If something's denatured, that means it can't go back anymore. So is that reversible? No, incompetitive inhibition is not reversible. Okay? You destroy it straight away. And what's coenzyme and what's cofactor, guys? Can anyone tell me? What's coenzyme? What's cofactor?
Something which helps facilitate the reaction of like ATP.
Not exactly.
Oh.
It's all right. Why it's called coenzyme? Why it's not called enzyme? What's the difference between an enzyme and coenzyme? Guys? Enzyme can accelerate the biochemical reaction by itself. The coenzyme can make your enzyme work better, more efficient. But the coenzyme cannot accelerate the chemical reaction by itself. Coenzyme is just like tutor. It's just like me, okay? I can make you study better, but you can't just study with me, okay? You have to go to your own schools. The teacher is your enzyme, okay? I'm your coenzyme. I assist your teacher. But I don't accelerate the reaction by myself. Okay. If it's called coenzyme, it's going to be made of what? Protein. Okay. If it's a protein that assists the enzyme to work better, it's called a coenzyme. If it's not a protein that can assist the enzyme to work better, more efficiently, that's called cofactor. Okay. That's the key point. If it's protein, it's coenzyme. If it's not protein, let's say metal or carbohydrate or so, or lipid or something, it's called cofactor. Okay. A coenzyme doesn't accelerate the chemical reaction by itself. It can only assist your enzyme to lower the activation energy. Okay. Done. All right. Let's say, okay, photosynthesis and cellular respiration. The disaster kind of as well. What's the difference between C3, C4, and CHEM? Nice.
C3 is like the chemical like trees and stuff.
Yes.
And having applications like a more environment.
A warm environment,
Other
like
environment.
temperate environment.
Uh,
Yeah, go for it.
C4 is for like other hotter environments. They have they separate the light independent stage by space.
Yeah.
And CHEM is they separate it by like
Okay, there's a few key words that you need to remember. If it's C3, it's just a regular, ordinary plant, right? Nothing special. If it's a C4, always remember spatial isolation. What does that mean? I do my carbon fixation and proper Kelvin cycle in different places. Okay, that's called spatial separation. So, where does your carbon fixation happen? Mesophyll. Yeah? You want me to go through it from the beginning? Our time is valuable, okay? Let's go through the general mechanism all over again. First step, light dependent stage. What happens at this stage? What do we need as a source? Water. Perfect. Anything else? Sunlight. That's the only thing that we need from the outside environment. Okay. What's the pigment that absorbs the energy from the light? Chlorophyll. Okay. Which structure within your chloroplast contains chlorophyll? Do you guys know there are different structures in your chloroplast? There is. Thylakoid, granae, and stroma. Okay. Which one contains chlorophyll? Your granae. Okay. A granae is just a stack of thylakoid. Okay. That's the thing contains chlorophyll. That means your light dependent stage only happen in your thylakoid or your granae. Okay. That absorbs the energy from the light. And what are they going to do about this energy? They're going to break the water apart into hydrogens and oxygens. And the hydrogen plus ions will be carried by the coenzyme NADPH, to form the NADPH, right? For the next step. And oxygen is going to form the oxygen gas. That's the waste for plants. They don't need oxygens. Yeah. So they just release it away. Okay. So the only thing that has been generated in your light dependent stage is your NADPH. That's the only thing that is important for your photosynthesis. Okay. But we know it's not just hydrogen in your glucose, right? What's the chemical equation for your glucose? C6H12. H12O6. That means you still need oxygen and carbon from it, right? Where are you gonna get those carbons and oxygens? Carbon dioxide from the outside environment, right? So in the light dependent stage, you don't need carbon dioxide. But for your light independent stage, you don't need, I know you kind of need water, you always need water. Well, water is not the main reactant for your light independent stage. The only thing that is important in your light independent stage is your carbon dioxide. That's the source of the carbons. The main purpose of photosynthesis is making organic molecules from inorganic molecules. The inorganic molecules is your carbon dioxide, organic stuff is your glucose. They're all just carbons. Yeah. So what's been generated? Before we enter the actual Kelvin cycle, we need to do something beforehand, right? That's called carbon fixation. Carbon fixation is always considered as your light independent stage, but not contained in the Kelvin cycle. Okay. So if you want to explain it very detailed, you can say light independent stage contains your carbon fixation and your Kelvin cycle. Okay. But a few years ago, you can just say Kelvin cycle equals to light independent stage, but it's not 100% accurate. Okay. What happens in the Kelvin cycle? What happens in the carbon fixation in C3 plants? An enzyme called Rubisco is gonna catch the carbon dioxide from the outside environment, right? And converts it into a organic molecule that contains 3 carbons. I don't know what's that called, and you don't need to know either. Okay, you just say C3 molecule. That's enough. That's more than enough. Okay, they use this molecule as a source of your Kelvin cycle. Okay, what can you put into your Kelvin cycle? That's C3 organic molecule. That's the only thing you can put in your Kelvin cycle. Okay, and after the Kelvin cycle, the glucose has been generated. Okay, that's it. Done. Easy, huh? You don't need to know the details of the Kelvin cycle until university. Okay. So, what happens in C4? What's the difference? And why there's a difference? Because C4 plants lives in a hot environment, tropical area. Okay. What's so special about tropical area? Guys, come on. What's so special about tropical area? It's hot, right? It's hot. That means the temperature is high. What's going to happen if the temperature is high? Your Rubisco will be more easy. It will be easier for the Rubisco to affiliate with oxygen rather than carbon dioxide. And if it is affiliate with the oxygen, what's going to happen? Photorespiration. That's the reaction that we don't want because it reduces the efficiency of your photosynthesis. Okay, photorespiration is something that we don't want during the exam, during the photosynthesis. Sorry, during the photosynthesis. Right. So we have to prevent it from happening. How do we do that? We don't want the Rubisco to see oxygens straight away. So we separate them by generating 2 different environments. One is mesophyll, one is bundle sheath cells. Okay, what's going to happen in the mesophyll cells? We're going to do the initial carbon fixation by another enzyme called PEP something. Okay, can't remember the name. Yeah, it's better over this. Okay, okay, and it's going to create what? It's going to generate another form of organic molecule that contains 4 carbons within it. Yeah. And this molecule will be carried to the next stage of your light independent stage, which is your carbon fixation, which is your Kelvin cycle. Can we put this molecule into your carbon cycle straight away? Can you? Why? because that C3 molecule is the only thing that you can put in your Calvin cycle. Okay, it's very picky. Calvin cycle is very picky. It only wants the C3 molecules. So what's going to happen next? In your bundle sheet cell, there's a Rubisco ready. Okay, so the Rubisco is going to do the second stage again to make those C4 molecules C3s again. Okay, so you can just think of this as a separate carbon fixation processes. The first step is making C4 because it prevents the Rubisco seeing oxygen in the environment. But the second stage is to make the actual source or the reactant for the Calvin cycle. Okay, and that happens in the bundle sheet cell. The actual Calvin cycle happens in the bundle sheet cell. Okay, so we still need Rubiscos, but just in different place and different time. Okay, make sense? Yeah.
What's
Okay.
the one carbon molecule called?
One carbon molecule. There's no one carbon molecule.
You know how it's a 4 and versus 3 and that one remains.
Well, you have, what's the common denominator of it? Like you have, let's say if you have 3 4 carbon molecules, and you can make 4 3 carbon molecules. Yeah. Let's say just something like this. Okay. You don't need to know the detail there. Okay. You just need to know the C4 converts into C3s.
I have an exam question that asked me what is the one carbon molecule named, and I was just like,
You won't be
that's what
assessed.
it's the carbon.
Did you see the actual question?
Yeah, on the exam I had yesterday.
That's a stupid question. Well, let's say carbon dioxide or something. I don't know. I think it's gonna be on BCA, I promise. Okay. If you're assessed, you can kill me afterwards. Okay. If you want. Yes, I'm always here. Okay. Yeah, what's next? C4 is explained, right? We need to prevent photo respiration from happening. What do we do? So can you guys know, do you guys know what's the temperature threshold for affiliation to oxygens for Rubiscos? If it's above 30, it's gonna affiliate with oxygen easier. Okay. If it's below 30, it's carbon dioxide. Okay. So what about CAM? CAM plants live in a hot and dry environment. What do we do? Can we open the stomata during the day? What's gonna happen? It's not photo respiration. What's gonna happen? You open the stomata. The water is going to evaporate drastically. Yeah? You have to prevent water loss. That's why you have to do it at night. Okay. Have you guys done unit 1, 2? Yeah. What's that called? The evaporation of water. Transpiration. All right. Transpiration. Okay. The water evaporate at the top and creates the pressure difference between the top and the bottom. That's why the root can absorb the water from the soil, right? All right. It's all right. It's not important. So, what's the main mechanism for CAM plants? They still use malic acid. They still have carbon fixation doing separately. Yeah. But the most important part in CAM plants is temporal isolation. What does it mean by temporal?
Separate light and reaction.
In a timely manner. Okay. So we do light dependent and light independent at different times. We do carbon fixations during the night, and we do light dependent during the day. Okay. What's the benefit of it? But the temperature in the desert is, well, much lower at night, right? So that means your carbon fixation will be more efficient. Yeah. And the stomata only opens during the night. That means the water evaporation can be prevented very efficiently. Okay. And well, they kind of use different cells, but it's not actual temporal, there's not actual spatial isolation. Okay. You just emphasize temporal isolation in CAM cells. Okay, that's the difference. One more thing we need to be careful with. During your light dependent stage, we're going to generate 2 ATPs. Are we going to use these 2 ATPs during the reaction or after the reaction? The 2 ATP that is generated in the light dependent stage will be consumed in your light independent stage. Okay, FYI. Okay, I don't think it's going to be on the exam, but just in case. Okay, you actually generate ATPs during light dependent, but there's no net output. Okay, for your photosynthesis. Okay, one more thing. Well, there's actually no one more thing. Okay, cellular respiration. 3 stages. What are they? Guys are so sleepy. Cellular respiration. Glycolysis is the first stage. Where does that happen? Oh, forgot that. Where does your light independent stage happens? In the stroma, okay? In the stroma for photosynthesis, okay? And the light dependence happens in the thylakoid or the granae, okay? And glycolysis in cellular respiration happens where? In the cytosol. The mitochondria is not related in this stage, at this stage, okay? So what's the energy output for this stage? And what generally happens at this stage? Oh, sorry. How many ATPs are going to be generated at this stage? 2. Okay, 2 net. We're going to generate 4, but 2 will be consumed. Okay, so we net output of 2 ATPs during your glycolysis. And what's the substance that we're going to create from 1 glucose?
Oh, 1 glucose to 2 pyruvate.
Pyruvate. Okay, pyruvate. We're going to generate 2 pyruvates. And what's next? We're going to get into the mitochondria. What's the next stage? It's called Krebs cycle. Yeah. What needs to be done before your actual Krebs cycle? Just like a light independent stage carbon cycle. There's something you need to do beforehand. It's called pyruvate oxidation. Okay. You convert 2 pyruvates into 2 acetyl-CoA. Okay, and that's the actual input of your light. That's your actual input of your Krebs cycle. Okay. And forgot to mention how many NADH is going to be generated in your glycolysis. 2. Okay, 2. The main purpose of the first 2 stages is just to generate as much of NADH as you can. Okay, that's the main source for your oxidative phosphorylation, ETC. Okay, where does that happen? Your Krebs cycle. Where? And you guys tell me the structure of the mitochondria. Has an outer membrane, has the inner membrane, has the intermembrane spaces. And what's within the inner membrane? Right, what's within the inner membrane? The really center. The matrix. Perfect. Yes, appreciate that. What's the foldings of the inner membrane? Okay. If there's no folding, this is called inner membrane. Okay. And in between your inner and outer membrane is called intermembrane space. Okay, very important in ETC. I think I've already explained last week. Sorry. Your second stage, your Krebs cycle happens in your matrix. And we generate as much of NADH and FADH2 as possible during this stage. Okay? Have we used any oxygen so far? Have we? Have we? Have we? Or you don't need oxygen to participate in the second stage either. Okay? But as a whole process, you will need it for the aerobic respiration. Okay? So what's being generated at this stage? I think you generate 8 NADH and 2 FADH2. Okay? Each NAD is going to carry 1 proton, and each FAD will carry 2 protons. That's more efficient. Yeah? But we don't have that much FAD in our body. Okay? So the main purpose is to generate FADH2 or NADH. Okay, and we're going to bring all of those stuff into your final stage, electron transport chain. Okay, so where does that happen? And how many ATP are we going to generate in the second stage? How many, guys? Just guess. Give me a number. You can say 100.
28.
2. Okay, you generate 2 ATPs at the second stage. Okay, second stage. So 2 for glycolysis, 2 for Krebs cycle. Okay, and we move on to the next stage, the final stage, which is called electron transport chain or oxidative phosphorylation. Okay, what happens there? Where are the protons located after we finish the Krebs cycle? No. Where are they? In the matrix, right? Because Krebs cycle actually happens within the matrix. Yeah. So what's going to happen next? Those coenzymes will convert or carry those protons to the next location. It's going to move it across the inner membrane. Just throw it into the intermembrane space. What's going to happen if you throw these protons that carries the positive charges into the intermembrane space? What's going to happen? There'll be more positive charges in an intermembrane space, right? So if we look at the voltage differences, it'll be more positive in an intermembrane space and be more negative, relatively speaking, more negative in the matrix, right? So that creates what? A voltage difference, right? And what's going to happen if you create a voltage difference? Go on, guys. What's going to happen if you create a voltage difference? You generate current, okay? You generate current. What's the main negative charges in our body? Sorry? Electrons. Electrons. Yeah, negative charges, electrons. If it's positive, it's protons. If it's negative, electrons. So once we generate the current, the electrons will move in a particular direction, right? And that movement happens within your inner membrane. Okay, even though the protons are located in the intermembrane space, the electrons is going to migrate along with the direction of your phospholipid bilayer. Okay, that happens within the membrane. And what's the special enzyme that is located at the intermembrane, that is located in the inner membrane, that is responsible for the production of ATP? That enzyme is used for synthesizing ATP. What's that enzyme called? Guys, are you guys listening? ATP synthase. Okay. Yeah, ATP synthase. Right? You guys actually understand how to draw graph then. And kind of. Here's your mitochondria. That's your outer membrane. That's your inner membrane. That's your intermembrane space. Okay. Where does the Krebs cycle happen? Here. Here's a matrix. Okay. Where's the electron transport chain? The foldings here. Okay. That's called the cristae. And if we zoom in, it'll be something like this. That's your inner membrane. Yeah. All the membrane are the same. Phospholipid bilayer. And we have this protein. Okay. That's the ADP synthase. Looks ugly, but yeah, you just bear with me. Okay. Here's the matrix. What's outside of the matrix? What's this? What's this? Intermembrane space, right? That's just outside of your inner membrane. Outside of your inner membrane, called the intermembrane space. Yeah? Between your outer and your inner membrane. Yeah? Sorry, where do we have those coenzymes carrying the protons initially? In the matrix, right? So NAD, H, and FAD, H2. Yeah? What's gonna happen? Yeah. They're gonna throw the protons across the inner membrane. So there will be a lot of H plus across the membrane there. Okay. So if we look at the charges in as a whole, it'll be positively charged, right? Something like this. Yeah. And in your matrix, relatively speaking, it'll be negative, right? Or you can just write it like this. That's the symbol for charges if you do chemistry. Okay. What's going to happen now? There's a voltage difference, so there will be a current being generated. Okay. Direction of flow, either way, I don't care. Okay. This way. That's the direction and the movement of your electrons. Okay. That's the movement of your electrons. You're going to move across or alongside with the phospholipid bilayer. What's next? It's just like a flow, right? If you generate a current, what's going to happen in the middle? What will happen in the middle? It's just like a waterfall, right? What do we do about the waterfall? We generate the electricity from it, right? Yeah. All right. Sorry. They carries what? Kinetic energy, right? Because they're moving. Or they carry charges, carry kinetic energy. So once they pass the ATP synthase, the ATP synthase will spin. Okay? Every circle, every 360 degrees it spins, it's going to generate one ATP. Okay? So it spins, ATP will be generated. Okay? But the number of protons that we carry across the membrane is limited. That means the current that we generate will be limited as well. Okay? So there won't be a lot of charges. So the spin time of your ATP synthase will be limited as well. Okay? And that's random as well. Okay? That all depends on the situation. If you're lucky, you can make a little bit more ATPs. If you're unlucky, you just make it a bit less. That's why it's not a fixed number in the electron transport chain stage. Okay, you generate 26 to 28 ATPs from this stage. Okay, 26 to 28. All depends on how many circles has the ATP synthase has spent. Okay. If it spends more, 28, spends less, 26. But generally around this interval. All right. If you're extremely unlucky, it'll be 23 or something, but highly unlikely. Highly unlikely. Okay, what's next? ATP will be generated. What do we do? What we're gonna do about those protons? If we don't do anything about it, the protons will accumulate in your intermembrane space, right? What are we gonna do? We don't want them because they've done their job already. What do you do? That's the time we need our oxygen in a chemical reaction. What's proton plus oxygen? What's hydrogen plus oxygen? Water, yes. So this is when the water is being generated, and this is when the oxygen is being consumed, okay? When does the carbon dioxide being generated? In your Krebs cycle, okay? Krebs carbon dioxide is created in your Krebs cycle. Water is created in your electron transport chain, yeah? So let's add them together. We generate 2 in glycolysis, 2 in the Krebs cycle, 26 to 28 in the oxidative phosphorylation or ETC. What's the total number of ATPs we're going to generate for each glucose? 30 to 32, okay? Simple math, huh? 2 plus 2 plus 26. Yeah. That's aerobic respiration. We can also have anaerobic respiration, yeah? What about it? What's anaerobic respiration? It's also called fermentation, right? It happens in 2 different types of cells. One is fungi, one is animal. Why plants don't do anaerobic respiration? Have you thought about that? Are you kidding me? They don't have mitochondria. Are you? Yes. Why they don't do anaerobic respiration?
They produce the oxygen.
Yes, they're the producer. They produce the oxygen. There's no way they lack of oxygen. So they always do aerobic respiration. Okay. If they don't have the oxygens, all of us will be dead. Okay. Sorry. When we talk about anaerobic respiration, fungi and animal only. Okay. What do we create for animals? Lactic acid and carbon dioxide. Okay. Lactic acid is just acid. Yeah. You'll feel extremely uncomfortable after 100 meters race, right? Because there's a lot of anaerobic respiration taking place. That means a lot of lactic acid has been generated. That's the acid, which means it's going to change the pH value within your blood. Yeah. And the pH value of your blood is strictly maintained, okay, to 7.45, strictly that interval, okay? Why is that the case? Why do we have to control the pH value of blood so strictly? Because there's a lot of hemoglobin in your blood, a lot of proteins. If you denature the hemoglobins, congratulations, you're dead. Okay. So we can do anaerobic respiration, but we don't do it all the time. Okay. Our first preferences is aerobic respiration. Okay. As long as we have the oxygens, we do anaerobic. Yeah? Okay, what about fungi? What about fungi? What do they generate? Do they generate lactate acid as well? What do they generate? Ethanol, yes. What's ethanol's colloquial name? Alcohol, yes. They generate alcohol. That's how we make wines, make beers, make spirit. Yeah. They make alcohols and carbon dioxide as well. Okay. That's how you make bread. Why there's so many holes in the bread? Because anaerobic respiration taking place and the gas fills the gaps in between in the bread. So that's and it's hard to describe. It's 发面, right? All right. That's about it. We've done everything before immunity. I think Joshua has went through all the immunity stuff with you earlier.
Oh, plasmid.
What was it?
Oh, plasmid. Okay.
Plasmid in proteins. What do you mean? What was it? Which page?
Oh, okay.
Insulin.
Oh,
Insulin.
Sorry.
What about it? Insulin is just an enzyme used for metabolism, right? Glucose adjustment. If you use I don't think it's on the syllabus anymore. It used to be on the syllabus. If you use insulin, you convert the glucose into glycogen and stored in your liver, right? And if you use glucagon, it converts it back to the glucose again. Right? Insulin, I don't think that's on the syllabus. Yeah, I don't think that's in the syllabus anymore.
Our deck.
Yeah.
Talking about that for 3 months.
Okay.
And then I lost.
Which school you got?
For.
Bowen.
And then-
Congratulations.
And then I lost 3 months about.
Congratulations. I can't do anything about it. Like back to my time, it was on the syllabus. But I don't think it's on the syllabus anymore. Guess. Do I look old? 15. That was a good guess. I'll end the recording. So not everybody can hear it. Okay, we've done, everybody. Okay, good luck with the exam. Okay, I'll upload the recording afterwards. Okay, you can go.